Well, good afternoon, everyone. Welcome to SADS Live 125. Great to get to be with you on this cold Midwest day here in Green Lake, Wisconsin. I'm Mike Ackerman, genetic cardiologist, and that's my dog Bella on cue. Every time she does this for us, it's not a ghost, it's just Bella. But I'm Mike Ackerman, genetic cardiologist at the Mayo Clinic and director of Mayo Clinic's Winland Smith Rice Genetic Heart Rhythm Clinic and the Winland Smith Rice Sudden Death Genomics Laboratory. And I bring you greetings from our CEO, Alice Laura, of the SADS Foundation, the Sudden Arrhythmia Death Syndrome Foundation. And so wherever you're joining us from across the globe, welcome. If SADS Foundation is your home because you have one of our conditions that we focus on to help families live and thrive, we hope we've been a good home for you. We hope that you find good information, great information, reassurance, confidence, hope, healing with respect to the journey that you're on and your particular genetic heart disease. And today is a very exciting program. But before we get into today's program, I don't want to forget that next Saturday, September 16th in Nashville, Tennessee, is going to be our next family seminar for the SADS Foundation, hosted at Vanderbilt University Medical Center and Vanderbilt Children's Hospital. And very excited about that. I wish I could be there, but you're going to enjoy that, those of you who are going to that seminar. And I want to thank the folks at Vanderbilt University, dear friends. One who's on our program today shortly, Dr. Prince Kannankeril, and his team will be providing great information to the families that come a week from tomorrow. There's still time to sign up, and you can see that on the side. Well, today's program, I've been looking forward to this for a long time, and we've looking at schedules, comparing times to get two people who are fantastic in our field of genetic genetic cardiology. They're very special individuals. They're good friends of mine, and I am very careful with the word friends. And so I mean that for Dr. Kannankeril and Dr. Abrams. So we'll have them on in just a little bit. But to set the stage, this is my wrapping up my 23rd year on staff at the Mayo Clinic. And for probably the first 20 years of those, we have not had any new therapies. We have never had an FDA approved drug with the indication being one of our genetic heart diseases. Well, the last 3 years, that has all changed and is changing, and it's incredibly exciting. For the first time ever in the field of genetic cardiology or genetic heart diseases that predispose to the possibility of sudden death in the young, we've had a first. That is an FDA-approved drug with the indication being one of our diseases, and that disease, hypertrophic cardiomyopathy, or HCM, and that drug, mavacamten, was the generic name. So it's starting to happen. And now what's starting to happen in the last 2 years is all kinds of clinical trials for drugs that are in development for our family's diseases. Not only drugs, small molecules, compounds, but also clinical trials for gene therapy. First patient recently received gene therapy for PKP2 mediated arrhythmogenic cardiomyopathy under the sponsorship of the company Rocket Pharmaceuticals. Later this year, first patients with MYBPC3 mediated HCM going to receive AAV9 gene therapy for that disease under the sponsorship of the company named Tenaya Therapeutics. In the drug space, there are new drug trials for patients with long QT syndrome underway, for patients with CPVT. And that's what we're going to talk about today, which is the state of clinical trials and what should families think about, what should patients think about, what questions should they ask as they're wondering, if I'm eligible, Do I want to be part of such a phase 1, phase 2, phase 3 clinical trial? What questions should I ask? And what answers, and depending on those answers, how does that influence my decision to do a trial? And so, well, I'm going to be joined now by Dr. Dominic Abrams from Boston Children's Hospital and Dr. Prince Kannankeril from Vanderbilt University. And they're going to help me with this discussion. And we'll go ahead and have Alice or Aaron bring these two fine gentlemen on camera. And drum roll. And on cue, we have Prince and Dominic, and I can call them Dr. Kannankeril or Dr. Abrams, but if you guys call me Mike, then we'll all go by first names if that's good with everybody.
Sounds great to me.
Will do.
Well, it's great to have you on SADS Live 125. It's not your first time on the show, so welcome back. And before we look into the state of clinical trials, Dominic, let's start with you with a reminder of your career journey thus far. Give our listeners an update on how you got from here to there so far.
Yeah, so as most people may already know or can hear by now, I trained in the UK. I was a cardiology resident at a hospital called Harefield Hospital, which was a transplant centre in the UK where Maggie Coob worked. And somewhat relevant to today's conversation, there was a patient who was referred down for transplant. He was 13 or 14-year-old boy, and this was 1997. And I only went to do cardiology because I wanted to be an anesthesiologist. They said to me, "You're too young. Go and do something else first. Go and get some more experience." So off I went to this cardiology. And this young lad, I was fascinated by his story. And I can't prove it, but I'm extremely confident he had Dannon's disease. And we did some biopsies, and we had this vacuolization seen in the cardiomyocytes in the heart cells, and so he had all the picture of it, had all the classic features, but we just didn't know what it was at the time. And in those days, a genetic test was just about impossible. So looking back, it was a fascinating case for me, and someone said to me, "You should write a case report about this." So excitedly, I went off with a resident's enthusiasm and wrote a case report that was about 80 pages long to begin with, which someone then pointed out they're not usually a bit shorter than that. But it was sort of in that reading and working around it that I became very interested in it. And then through my training in electrophysiology, I still work as an electrophysiologist, became very increasingly interested in heritage heart disease. And I've worked with some amazing people in the UK, Bill McKenna, Hugh Watkins, etc. And so it was a great opportunity. And then when I came to the US, the same thing here in the US, working with great individuals. So it's sort of been a journey. I've been very, I think, incredibly fortunate to sort of ride the wave of genetic cardiology as it's evolved as a specialty from this sort of rather rare, uncommon disorders that people weren't very familiar with when I started to now being much more mainstream. It's wonderful to see. And of course, we're here today to discuss new therapies that are now based on the genetic and the molecular understandings. Amazing place to be. So that's my sort of journey, and very excited to be here, a part of this conversation.
Great. Thanks, Dominic. And turning to you, Prince. From in the beginning, there was Prince, and then what happened?
Yeah, so I did not think I would be a genetic cardiologist. Sort of growing up, I thought I'd be an orthopedic surgeon like my father was. But then in med school, I really got turned on to pediatric cardiology. Really thought I was going to focus on congenital heart disease. I was actually a med peds resident at the University of Minnesota, and I remember hearing a grand rounds on these new genes that were discovered to be causative of sudden death. These new genes in SCN5A and these potassium channel genes. And at the time, that just sounded so bizarre and foreign. I didn't think I'd ever want to have anything to do with that. But anyway, I continued my training. I did my pediatric cardiology fellowship in Chicago, and then I came to Vanderbilt for my electrophysiology fellowship, and have stayed on ever since. And really, it was after being here and working with Dan Roden and Frank Fish that I really got turned on to the world of channelopathies. And so that's kind of where I've focused my career since then.
Great. And I know we're going to talk about clinical trials, but before we do, both of you have active research interests and programs besides your clinical practices. And why don't you go ahead, Dominic, and give a thought exercise to what's one question that your research team is really keen on solving or getting an answer to that you're excited about in your program or looking into?
So I think one of the things that we've been working on, one of the things we've been working on is catecholaminergic polymorphic VT over the last several years. And I'm part of a group. I'm very lucky to work with some amazing translational scientists. So we have a team approach where I do a lot of the clinical work and my colleagues Vasilios Bezaridis and Bill Pugh do the scientific work. And we sort of found that collaborating and working together has been very fruitful on that front. So I think, one of the things we're really interested in is we've got this potential therapy that works well in heart cells in the dish. It works well in mice. And then how do we transfer that to the human and get that into clinical trials? And I'm also very interested in the business aspect of it. Is now what do we have to do to make this work, and how do we understand that, and how do we get that forward? So that's sort of one of the things that we're really thinking about, is how do we get something successfully from looking great in the lab to actually being a reality for patients, because that's ultimately what we all want to do. We want to actually be able to sort of change people's lives and be able to effectively cure these diseases. So that's a sort of a big challenge that I'm really enjoying at the moment, and how we can do that. And obviously, as the science continues, then we hope that'll continue to move forward.
Yeah, thanks, Dominic. And how about Prince, for you?
Yeah, so there's a lot of just really fascinating questions in this field. Far too many to elaborate right now. But one of the areas that I'm really excited about, and spoiler alert, if you're coming to Nashville for the family conference, you'll hear about some of the work that Andrew Glazer is doing. He works with Dan Roten, and nowadays, genetic testing is fairly straightforward, but the interpretation of the genetic test is often pretty complicated. It's not uncommon for us to send off a test and then we get back a variant of uncertain significance, in which there's really no information on it. So what Andrew and his team are doing is they're doing high throughput functional testing, where they can now take a gene as big as SCN5A and make potentially every possible mutation, even mutations that have not been seen before in humans, and functionally characterize them so that hopefully in the near future there won't be true VUSs anymore. So when we get back a genetic report, even if it's never been seen in a human, we'll at least have some idea from the functional data that yes, this does or is expected to affect the QT interval, for example.
Yeah, there's so much exciting things happening, and the pace for which the questions can get asked and answered now is kind of incredible. And I know Dominic mentioned CPVT and potentially a very exciting new therapy that his team is advancing. And before we focus on what's happening right now, let's go down memory lane a second, Prince, because you were critically involved with one of the first, maybe the first, but if not the first, one of the first randomized clinical trials of a known drug established for other diseases to be brought to bear for CPVT. And most of our families now, they think, oh yeah, flecainide, we're all on flecainide. The treatment of choice for CPVT is combination drug therapy with beta blocker, Nadolol is the preferred beta blocker, propranolol if it's not available, and flecainide. And we all sort of do that as if that's the way we've been doing it forever. But forever is really only the last 5 years maybe. And take us back to what it was like launching that kind of a clinical trial. Not easy, and you did an amazing job. And now, because really that work of yours, flecainide is standard of care almost all in the vast majority of our CPVT patients.
Yeah, so that is a great story, and as you said, it's now considered standard of care. But back when we were starting, we really only had beta blockers, and then we would try calcium channel blockers in the severely affected patients, and they really didn't work all that well. So we didn't really have a whole lot to offer. And so it was, if I remember this correctly, it was around 2007 or so when there was the first signal that perhaps flecainide would be useful. And And that's actually an interesting story too. So Hiroshi Watanabe is a electrophysiologist who's from Japan. He was working here at Vanderbilt doing a postdoctoral fellowship with Dan Roden. And Bjorn Nolman, who's another one of the collaborators here, who's done a lot of tremendous work in a lot of areas, has a very robust mouse model of CPVT, the calsequestrin knockout mouse. And Hiroshi had heard about these mice and wanted to give them flecainide for reasons that nobody could really understand. There was no reason to think flecainide would be useful. We think of flecainide as a sodium channel blocker, and it is a sodium channel blocker. But anyway, Hiroshi, persisted and went ahead and gave flecainide to these mice, and it was, stunningly effective in this mouse model. And so what Bjorn did is he worked out the cellular EP and showed that flecainide also directly blocks the RYR2 channel. And of course, in CPVT, the primary molecular defect is excessive calcium leak through the RYR2 channel. So it seemed like it was, targeting the molecular defect exactly. And so shortly after that basic science report, Arthur Wilde in the Netherlands gave flecainide to a couple of his very severely affected CPVT patients. And so they made it into that first paper that was in Nature Medicine. And then working with one of his colleagues, Christian Vanderwerf, and colleagues around the world, we did sort of an open label trial of flecainide, where we, basically people who had CPVT patients who were not well controlled on beta blockers, we just added flecainide to them, and we saw a really good effect. But as we're going to talk about today, to really prove that a new drug or new therapy is working as we expect it, then we do these randomized clinical trials where we take patients and we assign the new therapy to half of them. We assign placebo to the other half of them, and then we try to demonstrate that those who get the new therapy actually do better. And in the case of flecainide, particularly for CPVT, we could do a very powerful trial design, and that's a crossover trial. So every patient actually received flecainide as part of the prospective trial, and then they all crossed over to placebo. So they were taking a drug that looked and tasted like flecainide, but it was just it was inactive drug. And we randomized the order of those therapies. So half the people got flecainide first. We had them exercise, and then they were treated for a period of 18 months, and then they crossed over. And so using that, like I said, that crossover trial is very powerful because all the interpatient variability is sort of removed because you're comparing each patient directly to themselves. And so with a relatively small number of patients, we could show pretty definitively that flecainide made the exercise test look a lot better. It turned out, the trial, ended up not being powered enough to show a difference in clinical events because we had so few clinical events during those 18 months on each therapy. But we did in fact show, as you said, in one of the few randomized drug trials in genetic arrhythmia syndromes that this new drug was in fact effective compared to placebo.
Yeah, and I think what's interesting, you mentioned about the drug normalizing, improving, treating the stress test, making it look better, but not powered enough for event rate reduction. And that's really kind of that in a previous study with eleclazine in long QT syndrome was kind of a good news for all of our families out there, because the FDA has up till now accepted improvement, shortening of the QT interval, or improvement, removal, reduction of ectopy on the stress test as acceptable endpoints for efficacy, rather than needing the trial to be powered and then take decades of time to ultimately show event rate reduction. Because as clinicians, we all know that anything that shortens the QT interval is good for our long QT patients. Anything that removes the ectopy on the stress test is good for our CPVT patients. So I think that's been good news that many people don't know, is that the FDA really embraced that forward-thinking approach so far, as accepting those endpoints as surrogates of efficacy. Any follow-up thoughts on that, Dominic, before I ask your thoughts on some of the trials and what families should be asking?
Yeah, I think that's incredibly helpful from the FDA, because we need to understand what are meaningful endpoints. And so being able to address those, being able to shorten the QT interval, suppress ectopy, is very important, and having experience of that in already is going to be very beneficial, I think, going forward in understanding what are truly meaningful endpoints. And I think that, for many things, these can take a long time to come to fruition, particularly if you're looking at events as endpoints, which often can be quite well spread out. So being able to demonstrate short-term endpoints that show efficacy of a therapy, I think, is really, really important. And it's good that the FDA recognized that and that we have a precedent in place before we move forward with new therapies.
Speaking of new therapies, let's look at those. So we have gene therapy on the horizon starting to happen. We have Thrive Therapeutics with a new long QT drug that's enrolling patients soon. We have 2 different CPVT clinical trials, at least 2 that I know of, that are enrolling patients. And I'll do the easy part first, and then ask you, Dominic, to comment on some of the harder parts. So the easy part for all of our families is this answer: no children are eligible for any of these studies right now. So a lot of the families are asking, got an email this morning about where's gene therapy for my baby. So right now, for every single trial that I just mentioned, all trials are for those individuals with disease age 18 and older. And that's because that's been the requirement forever of the FDA, is that these trials and these therapies need to get established with their efficacy in the adults first, and then the children, unless that disease is a pediatric exclusive disease. Then you can start the trial in the kids. But so far, if you're under 18, all of you get a watch with great interest as to whether the trial is successful or not, but we will not be able to enroll your son or daughter, your grandson or your granddaughter in these trials. For the rest, Dominic, that are 18 and older, and maybe I'll have you do the long QT one. I know you are involved. Maybe a two-pronged question. One, when you and I and Prince decide to serve on a scientific advisory board for these companies, and maybe it's even the new company that you're now involved with your new compound, what do you look for in that company, in that trial, that would make you comfortable to add your name and reputation as an advisor or to be involved with said company?
I think you want to look at I always want to look at the bigger picture. So firstly, always saying, how good is the science? Does the science look convincing? And, for long QT3, for example, is this a molecule that's going to be effectively work against the molecular problem that we see associated with long QT syndrome type 3? So does the science make sense? And then I think importantly, what is the potential for this new medication to get to market, and what is the market? What are the number of people who are going to potentially benefit from this? So sometimes there's some amazing science out there, but there aren't enough patients that we can access or we can get to. And I think it's really important that the companies also engage with the patient communities to understand what are the issues that really face them. I think as sometimes in medicine we think we know, but unless we really truly ask what the problems are, it's very difficult to understand that from the patient perspective. So we really need a detailed understanding, and bringing the patient community in early, I think, is hugely important. So really sort of understanding, does the science, does the potential treatment benefits align with what the specific problems the patients have? That's a really important thing. And then I think just looking at the company, what are the who are the founders, what are the bases of the company, and just sort of understanding more about it. And obviously something that myself or Prince or others or yourself are passionate about. It's something we really would like to help with, be part of, and contribute our experience as best we can to try and make this an effective treatment. And it's a great pleasure and an honor to be in that position, to be able to do that. It's a really, really nice thing. And I think, it's a great opportunity to have some honest conversations about how we can really help. So that's a sort of a broad overview of what I would look at in thinking about how this could work.
And then the Thrive Therapeutics one, and full disclosure, both Dominic and I are on the advisory board. We're involved with helping that company with the clinical trial. Proprietary assets from Mayo Clinic have helped iterate the final drug that they developed that is being tested in the patients. So with that said, why would a family with LQT2 or LQT3, and that's where the trial is starting with for the families out there, 18 and older with LQT3 or LQT2, let's say they're study eligible, their QTC meets the enrollment criteria, and they would be eligible. Why would they want to do it? Why does the community need those eligible patients to choose, or at least enough of them to choose to be clinical trial subjects? And what should those eligible patients be thinking, asking, doing?
Yeah. So I think it's very important in looking at a clinical trial is what are the potential benefits to me as a patient that are over and above the current standard of care. So we know many patients, particularly with LQT3, have significant foundation. They can be more arrhythmogenic. The current therapies that we have, beta blockers, maybe mixelatine, aren't as effective as, for example, beta blockers are in LQT1. So is there an unmet need for me as a patient to improve my quality of life by taking a new therapy? And is this new therapy going to be better and supersede the current treatment strategy that's in place for me? So if a patient who's having regular shocks with an ICD, that is a hugely diminishes their quality of life. You never know what's going to happen. There's so many factors that go into that. So a new therapy is very important. And clearly it's very important that patient is eligible and qualifies. So of course, clinical trials tend to attract early people who, where the need is greatest, that they obviously are going to have more investment in the trial and be more enthusiastic to be part of it. But we all take a very different perspective as individuals as to whether we want to be involved in the trial at the beginning, sort of an early adopter, if you like, or whether we want to just sit back and see what happens and assess things more over time. So I think it's. Oh, Mike, sorry.
Yeah. No, I know. Thanks, Dominic. Yeah. And Prince, your issue with CPVT now is kind of interesting, isn't it? We went from having no new trials, no therapies, nothing, to now our patients with CPVT don't have one potential clinical trial to sign up for. They have two that are in motion or in operation. How do you decide which one you may want, or maybe there's different eligibility criteria, and that's how you as the enrolling physician might look at that? But how do we help families with CPVT answer the question, which of these two CPVT trials do I want to do? Should I do? How do we go about that now that we've gone from nothing to multiple?
Yeah, no, that's a great question. And as Dominic said, the trials tend to lean towards, the more severely affected patients. So if you're on maximal therapy and still having problems, yeah, you're going to sign up for whichever trial you can get into first, probably, and if that works, great. And if it doesn't, you'll try the other one. But for the people that are not so severely affected, let's say they're doing reasonably well on current therapy, they might ask, well, why would I, try this new trial if I'm doing okay? And there's a couple of reasons. One is, current therapy for CPVT specifically, beta blockers plus flecainide, and beta blockers are relatively safe, but plenty of folks have side effects. And I'm predominantly a pediatric EP, but as people get older, they tend to tolerate their beta blocker less well. I'd say my adult colleagues see a lot more beta blocker intolerance than I do. And then flecainide, there's this famous trial, the CAST trial, that actually showed an increase in mortality with flecainide in patients that had essentially ischemic cardiomyopathy. And so, as our CPVT patients get older and they get, coronary artery disease, they may not be okay taking flecainide for the rest of their life. And so it's important to have other therapies, that you can use as a backup. And these new therapies, frankly, might be, easier to take than natalol or flecainide. Flecainide, you've got to take it at least 2, if not 3 times a day. So once a day medicine is just easier. It may have a better side effect profile, and so those are important reasons. And then of course there's the general scientific reason is, yes, if you are able to travel to these sites and be a study subject, you could benefit all your colleagues with CPVT that don't have the wherewithal to be a study patient, and that's very important for the field in general.
Yeah, the field really needs patients, and not everyone, even if they're eligible, has the right mindset, if you will, right, to be a clinical trial subject. But the field, obviously, and the diseases need enough of those kind of patients who want to be a clinical trial subject in order to ever get smarter and develop new therapies. And to follow up on that, Prince, Mike is asking, is it common or even possible to participate in a second or more clinical trials for the same condition after completing one, whether the same or different modalities? And I guess that fits with the fact that CPVT, because we have active enrollment from 2 companies happening right now. I know at least for one of them, Armgo Pharma and their CPVT drug, the first patient in Amsterdam received it, and we have a second patient at Mayo who is screened eligible. So that trial is starting. I'm not sure of the status of the other CPVT trial. Maybe you are, Prince, and you could update the listeners about that. But the question of doing multiple trials, is that possible?
Yeah. So, and just for full disclosure, I'm not actively on any of the advisory boards for these companies, so I actually don't know what the status of that second trial is. Most of the time, you cannot be in 2 trials simultaneously. With very rare exceptions, they pretty much restrict you to being in that one trial while you're on it. Now, when that trial is over, when you're completely done with that trial, you can certainly, in most cases, do additional trials. So obviously, anybody who is in the flecainide trial can be in any one of these trials that are going on, right now or coming out in the future. Many times, particularly when there's a new drug, after the official trial is over, there'll be a post-trial follow-up period. So if you end up staying on that new drug, you'll often be approached to enter into sort of a registry or a follow-up study, in which case obviously they'll want you to stay on that new drug and probably not try any additional new therapies. So that's sort of the answer to whether you can be in more than one trial or not.
And sometimes families are bummed when they get excluded. And Dominic, could you comment on that? So like Jeff is asking, is there an upper age limit for PKP2 gene clinical trials? And we'll extend that, just the observation that the trials are carefully designed. The inclusion criteria are crisp and sharp. And sometimes somebody may really, really want to be in the trial and they're told they can't, and they're a bit bummed about that. Talk about that a little bit.
Yes, that's a great question. So I think that, as you said, Mike, for all these clinical trials, it's really identifying which patients are going to benefit the most and who is this really going to be appropriate for, particularly at the beginning. So when it's still in a trial phase, who's going to benefit the most? I think so many of these things come down to personal decision. Many people, I see a lot of adults as well, and they sort of get to a certain age in life and they think, I don't want your treatment. I'm fine. I've lived to 75. Why would I need anything else? Other people think very, in a different way and do want treatment. So I think these things become quite personal in terms of what specific people would like to do. Would you like to be involved in trials? Certainly, I know there's obviously there have been lower age limits at 18, which for something like PKP2 isn't so much of an issue because we see this much more in older patients, but of course we do see it in teenagers, and in many ways they may be those who could benefit the most. But I've never seen sort of specifically older trial limits, but it's important to understand how the human physiology is going to tolerate with something like gene therapy, which at the moment is still a big viral load and a big immune or big hit to the immune system. So it's important to think about those things, and also the medication you need, which may be very different when you're later in life than you are earlier in life. So I think it's all important to understand what are the potential benefits versus the potential risks, and it's weighing those up for any one individual.
Yeah, and Rachel, Dominic, is wondering, what are the things that exclude you as an adult? And the short answer is there's a lot of things potentially, but what are some of the things that you've seen in clinical trial design that your disease might make you eligible, your QT number might make you eligible, your stress test ectopy, if you have CPVT, might make you eligible, but you are not eligible because this, and this. What are some of those thises?
So I think all the this is are sort of very carefully thought out. Naturally, people designing the trials are going to be very cautious because the last thing we want is to have any harm to anyone. So, for example, some new therapies, if you're trying to have a family or planning actively to have a family at that time, that would be something that would be best avoided while you're going through that. So there's many different things. And often it depends on how the agent functions in the body, how it's eliminated, renal function, liver function, all these kind of things. So there may be other medications that you're on that may interact. And if those medications are essential, then that's going to be a problem. So it's really trying to make sure that the one individual is totally suited for this trial and that there aren't going to be potential harmful effects that may outweigh the benefits. So.
And I think I know the answer, and I think the three of us are going to be unanimous in this. But Prince, a lot of the families are learning of some of these trials now, either through SADS Foundation or through directly from companies. And they may be aware of these trials before their local physician is aware of these trials, because oftentimes the families might be farther ahead than some of the local physicians unless they're at an expert dedicated genetic heart rhythm center like the three of ours. Do these families need to tell their physician that they're thinking about this trial, wanting to enroll in this trial? Do they need that local physician's permission? What's your recommendations on that?
Yeah, it's always best to discuss it with your local physician and just inform them. And again, if they're completely unaware, most of them will still be very interested to learn about the study. So I know I've had requests from physicians, can I see the study protocol? And have the study team will always happily share that with the local physician, and then you can have your local physician review it for you and tell you, hey, here's what I like about this, here's what I don't like about it, for you specifically. So yes, I would absolutely recommend that you discuss it with your local physician, even if they hadn't heard about it before.
Thanks, Prince. And Dominic, I'm going to give a last one because you gave us a teaser at the beginning that was pretty exciting about the future question that your team is actively answering. And we mentioned 3 trials plus the gene therapy trial. So there's probably right now in your and my world, I think at least 7 trials that the three of us are needing to be thinking about whether our patients are eligible for to enroll this year. What about the next around the corner one, as in the one you're working on?
Okay.
What timetable do you guys see yourselves on in this from mice to man or mice to person translation? What's the next steps in its evolution? Because that's pretty exciting, and I know our listeners and Prince and I would be keen on the latest, greatest update.
Sure. So just to sort of give people a very quick overview of what this is about, this is a trial for CPVT, and it's known that there is a molecule downstream of the ryanodine receptor called calmodulin kinase. And if you can successfully, it's been known for some time that if you can successfully inhibit calmodulin kinase, you can in fact really suppress the arrhythmias associated with CPVT. The big problem of giving a medication to do that is that CAM kinase is expressed across the body, particularly in the brain, and is involved in things like memory and important functions that we all need on a day-to-day basis. So what my colleagues Vasilios Bezarides and Bill Pugh created was a cardiac-specific calmodulin kinase inhibitor. So it has a cardiac promoter, that means it just affects the heart, and they've been able to inhibit that, as I said before, in pluripotent stem cells taken from patients with CPVT who were very kind to give us blood cells, and then also from mice who were created to have CPVT. So that's now moving forward. It's moving into larger animals. There's a lot of collaborations going on, and I think it's a very exciting time in gene therapy. Like a lot of other gene therapies, there's a lot of work around what's the best vector to get this into the heart, what's the most effective, and what can do the sort of the least or have the least effect on the immune system, which remains a big issue. Even though these viral vectors are attenuated, there's still quite a big immune response. So things are moving forward. There's a lot of discussions that I'm not part of, so I can't expand too much, but sure. We're excited about it and hope that it continues. And I think it'll be very interesting to see where this goes, particularly if there's other can-mutation kinase inhibitors as well, and what's the potential benefit of it. So still early days, we're still preclinical, but hoping that it will be not too distant future.
Well, all the best to that. And just for the audience and the listeners to know, for the 5 trials or however many are enrolling right now, there were probably 20 that people were excited about in the last 3 to 5 years, and those went belly up. So that's the world of research, and you so far so good on the team in Boston, and Prince and I are going to be very excited about that, Dominic, and happy to help when it comes time. But I think to close, one of our challenges in CPVT, isn't it, is it's Prince's fault because of flecainide and because of nadolol flecainide, and because in the past we kind of accepted some ectopy to hang around on the stress test, whereas I would say now most CPVT centers, our therapeutic goal is to cure the stress test, to make the stress test look completely normal. And we are doing that with nadolol and flecainide, or denervation and flecainide, or triple therapy, denervation, nadolol, and flecainide. And as a consequence, a lot of patients with CPVT are not study eligible because you need enough stress test ectopy to show that the new kid on the block, the new drug, is working. And none of us really want to subtract therapy when we've cured the stress test to let them try the new therapy. So we're kind of, Prince, I'll give you the last word. We're kind of in this unique phase where it's getting harder and harder to find some eligible CPVT patients because on current therapy, their stress tests are looking too good.
Yeah, no, that's the huge challenge, because again, you'd have to then say, yeah, I'm gonna replace something that I know works with something that I don't know if it works, right? And so a lot of people don't want to sign up for that, and then a lot of studies aren't set up to do it that way. So you're starting with only those that have residual ectopy despite both medicines. So you're only looking at, again, the most severely affected patients. If you can then show that it works in those after you've completed that study, then perhaps you can talk about replacing one of these other ones. Again, as long as the overall risk benefit profile is better for the new drug than either one of the others.
Yeah, it's a good problem for our patients to have. That current therapy is really, really working well. But I think one of the exciting chapters that we're going to eventually get to is quality of life improvement. Yes, we can make the stress test look cured on current therapy, but can we do better than that in terms of taking care of the whole person? Yes, mexiletine shortens the QT and the LQT3 person, but they take it 3 times a day and it upsets their stomach. Can we do better than that? And I think all of us are hungry and eager to continue to find ways to not just treat the disease, but improve the quality of life so that we add to that mission of the SADS Foundation, not just living with your condition, but thriving despite your condition. And so I think we're in a good place. Our families are in a good situation to be dealing with their diseases now than, say, 20 years ago, when we look back and our therapies were pretty primitive just 20 years ago and have really come a long way. And so with that, Dominic, Prince, thanks so much for taking the time out. I hope I can have you come back on another SADS Live on any topic, but maybe when it's right, Dominic, for a progress report that you're eager to share with yours, and Prince with a progress report on making interpretations of genetic test results much less vexing. That'll be a fun progress report to discuss. So I'd love to have you back. You two are among my favorites. So, and that is now out for the world to hear. But have a wonderful weekend, Dominic and Prince, and to all of you out there in SADS Live land, all the best. Have a great weekend. Some of you in Nashville have a wonderful conference in 8 days, and the rest will see you back for SADS Live 126. Take care, everyone.
Thanks, Mike.
Thanks, Mike. Bye.